All right, in the beginning of chapter 1, we're going to be looking at the scientific method. We'll go through an example as well and look at the difference between science and technology. Let's get started. This is a drug that we're going to talk about. Taxol is still a current drug for cancer, specifically I think breast cancer. And just to give you a little bit of background information, back in the day, okay, for many years it's been known that a lot of stuff from nature can be used to treat different ailments. Willow bark, they would chew it for salicylic acid. This is even before they knew, oh, it contains salicylic acid. Okay, so salicylic acid's anti-inflammatory, used for pain. So knowing this stuff, teams of researchers would go out and try to screen plants. So back in the 1950s, National Cancer Institute said, let's screen some plants. So some group of scientists went, extracted plants. Another group of scientists then would try to do some tests. And then it was found that there was, in the specific yew, the bark of it, something in there would inhibit tumor growth. Okay, so another group took this and actually was able to isolate the active ingredients, paclitaxel. They found its formula later on, okay, so years later, a couple years later, they determined its structure. And then you can see, based on this timeline, it took even more years before it went to some clinical trials and got approved by the FDA. Awesome. Okay, here's a drug that works for some cancers. There's a problem though. When you take the bark of the yew, okay, this yew tree, the bark contains only a very small amount of the active ingredient Taxol. And when you take the bark off a tree, guess what happens to the tree? All right, so here's the problem. It's not a good drug choice because you have to kill trees, okay? And you need to kill thousands of trees just to give a small amount of Taxol. And if you have one patient, you would have to kill 6 100-year-old trees. So even though this is great, you could see the expense of it would be really high. And you could also see that lots of people would be fighting over trying to get these trees. And since they take so long to grow, it's not really a great renewable resources that you can get them right away. So another solution is needed. How does chemistry do this? Well, okay, looking at what that ingredient or the actual chemical formula for the Taxol was, can they find something else that's similar? So of course they're going to try other trees, and they found that the English yew, okay, which is a common tree and it's very fast growing, its needles, okay, and you can harvest needles without killing the tree, has a very similar drug or compound to the Taxol. And with a little bit of work to it, they can turn it into the Taxol and get it to do the same and actually even maybe a little bit better job in being an anti-cancer drug. Okay, Taxol hinders the cell division. It gets kind of stuck in there and doesn't keep it from dividing, which you would need. Cancerous cells have to divide very quickly. And so if you're preventing that from happening, you have now a great drug to kind of stop the cancer. Couple things to note in here. If you looked at the previous timeline, things like this in chemistry don't always move fast. It's not like one day we discover a cure and the next day everybody is now using it. It takes a lot of time before things are discovered, isolated, improved upon, and then also then getting trials and going to the market. Long time in there. And also there's lots of scientists at work. There was a group that kind of isolated it. There was a group that did the other work. So it's a very collaborative process as well. So looking at scientific method here, okay, you can kind of see there are so many images for the scientific method if you searched online. This is just one. And the main thing I want to show you is, you're starting with a question up top, okay, and there's always some background information that's done, or you have to look at what other people have done in that field already. And then you can come up with a hypothesis, your best guess. And of course, those hypotheses have to be tested with experiments. You need to collect data to show that you can make some conclusions based on that. And then here's the part where it kind of goes in a loop, okay? Oftentimes you're getting data, which then raises more questions for you, and it might change your hypothesis, and then you're going back and doing more experiments and collecting more data. Now, once you get some data collected and you can kind of explain it the best you can, you need to always communicate your results, okay? There's primary research literature that's been peer-reviewed, okay? Peer review, we'll talk again about this as well, but this is just a way that you write it down, it's reviewed by your other peers, so other scientists as well, to make sure that your experiments have been set up correctly, that your results have been drawn correctly. And so once it gets in there, it should be a very trustworthy source. But the great thing about chemistry and about science in general is you discover something, okay, later on you might discover, oh, hey, maybe there's some changes, or we didn't do this test and didn't think about that. So it's an ever-going process. Just some definitions here. Observations, you see things, you're observant, and you can make a hypothesis. Hypothesis, your tentative idea to explain your observations, and you have to be able to test it, okay? So further experiments must be done. So for the Taxol plant extracts treat cancer would be the hypothesis. So then you need to go and actually do some experiments on the plant extracts to see if that actually is true or not. Your experiments will generate data, okay? It could be both qualitative or quantitative. Qualitative, that means there's no numerical information in that data, okay? So it might be a texture or a color or something. That would be qualitative, and then quantitative is data that does contain a number. So here is some data. Extracts from Pacific yew bark kills cancer cells. Would that be qualitative or quantitative data? Is there a number there? No number, so that would be qualitative. And then compound 13A, okay, one of the ones they isolated, is 20 times more effective than paclitaxel in killing ovarian cancer cells. You have that number. They have results that show it's 20 times more effective. So this would be a quantitative. All right, a little bit more definitions here. When you have a bunch of experiments, you have a bunch of data, you can then take a law, make a law to kind of summarize those experiments. You have heard the law of gravity, the law of thermodynamics, okay, so energy can't be created or destroyed. Those laws are summarizing experiments that have been done and shown to be proven true. A theory then is taking a bunch of laws and experiments and will explain them, okay, so it's a unifying principle, so keyword there, that the theory explains a body. So the law just say, hey, this is this, straight up. This means this, it's just very clear cut. And a theory then would be more of an explanation. Students will say, well, it's just a theory, okay, when you get to some big ones. But theory in chemistry or in science in general is a pretty solid thing, okay? It's not contradicted by known experiments. And you can use theories to then predict unknown results. And theories can be disproved in the future, okay? They're ongoing. That it's always open. More results can then cause you to adjust your theory. But theory is a powerful thing. You don't ever want to say, yeah, it's just a theory. All right, last slide here, science, technology. All right, so science, okay, this is our experimental, okay, when you're investigating and trying to explain the world around you. And then technology takes that, what you have learned in science, and see how can we apply this? What is useful about it for us? All right, so science is necessary. It kind of is your pipeline to technology. Your life is so affected in everything you do from the time you're up to while you're sleeping by the technology all around you. So some important questions for all scientists to ask and for people, because guess what? Your tax dollars are paying for science research, which is a great thing. Is science always right? Okay, where are you getting your sources of your science? Technological advances, are they always good? And sometimes things that we think are good as we continue on, later on turn out not to be as good. Examples, there used to be lead paint, okay? Lead was an additive to paint. Lead was also an additive to gasoline. Came out later, we realized, okay, putting lead in things where it could get into your body and your system, probably not a good idea, and we have removed those. All right, so there's always things that sometimes you go forward, you realize you have to go back and fix something. And that's just part of science and technology. What should be studied? Okay, where is that research money, your tax dollars going to? Something where you should be engaged. You should be reading, looking at primary sources, and making good decisions. Bias, okay, you're always going to have, if you have an article and you read it and it's just trying to scare you or getting you to click on it, you probably want to check the sources and go to a primary source. Lots of stuff, you could do lots of great introduction stuff with science and technology and how they intersect. And we'll continue to hopefully get you excited about chemistry as we go on in Chapter 1.